Hi, everyone. I'm Shannon Wyman, the Scientific Communications Manager at Keystone Symposia, and I'll be hosting this Meet the Editors Roundtable today. I have with me today editors from Cell Press, the Journal of Cell Biology, Nature Communications, and PLOS Journals to discuss the state of the field, exciting new directions in neuroimmune and neurodegenerative disease research, as well as tips on how to publish in these top-tier journals. Before we get started, I'll go through some instructions on how we will run the sessions today. Please keep your camera and microphone off during this first portion of the event. We will start with a 30-minute discussion with our panel and then open up the discussion for audience questions, at which point I will ask you all to join us on screen for the interactive portion of the event. You may submit your questions at any time into the Zoom chat. During the audience Q&A, I will select questions from the Zoom chat and ask the person who submitted the question to turn on their video and microphone to ask the speakers your question live. After asking your question, please turn off your microphone once again. Okay, now I think we are ready to welcome our editors. If our editors could please join me on screen. Great. So to get started, I'm going to have each of you briefly introduce your journal and your role there for our audience. Swapna, you want to get us started?
Sure. So thank you very much for having me. My name is Swapna Ramu, and I'm an editor at MED, which is the new flagship clinical and translational journal from Cell Press. We launched in December of 2020 was our first sort of mini issue, and then our first full issue was in January 2021. So we really haven't been around that long. Obviously, as the name suggests, clinical and translational scope means that we are broadly interested in a lot of different things that have been discussed at the meeting so far, and will continue to be very much focused, of course, on therapeutic relevance to human disease, and clinical potential. So I think very broadly speaking, that is what we would be looking for at MED.
Great. Tim, would you like to go next?
Sure, thank you. Thank you again, Shannon, and thank you to the organizers for having this session. Thank you for inviting me. My name is Tim Spencer. I'm the executive editor of the Journal of Cell Biology. I think many of you have at least heard of the journal if you've not necessarily read it regularly. I think we're at the opposite end of the spectrum. We are quite an older journal. We've been around since 1955, so we're in our 66th year of existence. And as the name implies, we're interested primarily in work that is related to cell biology, cell and molecular biology. That's not to say that we don't also publish things that are, that have behavioral aspects, or that have, we're talking about neuroscience circuit level or other things. But essentially, what we're primarily focused on and what is of most interest to our community are things that are related to cell biology, the inner workings of cells, and how cells collaborate to essentially modulate behavior or disease, as the case of this particular, symposia. So our papers tend to be a little bit more mechanistically focused, but we certainly have a broad range of topics that we cover in our journal.
Great. Sophie, would you like to go next?
Yeah, hi. So my name is Sophie Morgan, and I work for Nature Communications. So Nature Communications has been around for 10 years. We just had the 10-year anniversary, and it's a fully open access journal. But we publish all sorts of natural sciences, not just neuroscience, but we have a neuroscience team. So I'm in the neuroscience and psychology team. But we publish a very broad range of neuroscience. So we range from all the way from molecular neuroscience up to like the psychology. We do computational systems, and also neurogeneration and neurological diseases as well. And yeah, so we'll kind of be looking for, I guess, for this audience, kind of neurodegenerative, broad submissions, like nothing too specific, kind of very broad journal. And yeah, broad scope as well.
Great.
Not too focused.
And Gabriel.
Hey, I'm Gabriel Caskey. I'm a senior editor and a team manager at PlusBio. And as the name of the journal says, we're a very general biology journal. So we publish from crystal structure of proteins to ecosystems and everything in between, including immunology and neuroscience. I handle mostly neuroscience papers, and we're also a journal that focuses on mechanism of biological processes, but we have other formats. We publish short reports, which are more conceptually novel and probably not fully developed story. We publish methods and researches. And in either format, we publish some translational research as well.
Great. Well, to kick off our discussion today, I'd love to hear from each of you about what do you think are the most exciting advances you've heard so far at the meeting or are looking forward to hearing about in tomorrow's sessions. And anyone who wants to go first, just jump in.
I'm happy to start.
Great.
So, I mean, actually, so far I thought the meetings, both symposia have been great, fantastic. One of the things that I'm personally excited about is, in part for selfish reasons, you know, coming from my background in cellular molecular neuroscience, I have always been interested in the sort of identification and, you know, of cellular subtypes, particularly glia, including regional subtypes, understanding their function, their specification. And this is something that has been sought after for quite a long time, but we're finally getting the tools now to really dive into those questions, which I think is really fascinating. And I think that's just, you know, sort of, we've only kind of hit the tip of the iceberg. And there's already been some great talks about cellular subtypes, particularly microglial subtypes, astrocyte subtypes. I think this is really fantastic stuff. And one of the things that I kind of is sort of interesting to me, because I've been coming to this meeting for a while now, or is these, I believe that these two meetings have been held, you know, concurrently for a few years now. Mm-hmm. And each time I go, they tend to be sort of converging closer and closer together. You know, they can almost merge into one meeting, which I'm not recommending because right now it's great. We get two sets of talks for the price of one. But I do think that they're sort of converging in terms of the subjects that they cover. You know, you can go to the neurodegeneration meeting and see talks about glia. You can go to the NeuroImmune meeting and see talks about neurodegeneration and immune interactions. So to me, that's fascinating. And I think, you know, from the journal perspective, there's quite a lot of work out there that is very cellular molecular that would be great for our journal.
Yeah, I think you're definitely right there. NeuroImmune used to be kind of a little satellite field off of this large neurodegeneration area, and it's really taking on a world of its own now. Yeah,
I do believe that. I think, particularly when it comes to the neuroglia interactions, I think Ben Barra has pulled the neuroscience community kicking and screaming into the 21st century.
Great. Great. Anyone else?
I suppose I can go next. Mm-hmm. So again, sort of informed by my background in lymphatic biology, I really enjoyed all the talks that are talking about, you know, the meningeal lymphatics, their role in normal function and disease, the various immune cell populations that are found in this region, how they're all interacting. And I'm also really enjoying how these are being escalated to the level of behavior. We heard a little bit of that today, and I think we have more talks tomorrow that are going to talk about some of the behavioral aspects. I think it's really exciting, and it's also very elegant to go from a single cell and looking at transcriptomic changes all the way to influencing something as complex as a behavioral outcome. So that's really exciting. And the other thing I'd like to pick, of course, not surprisingly given the journal I'm from, is the session that had a lot of industry speakers yesterday. I think that was really useful and also a sobering look in some ways, that you have all of these really brilliant biological discoveries, but they don't necessarily translate to actionable insights into the clinic. You have people that put in a lot of time and effort, and then you don't get the results that you want. So I think it's always really useful and important to keep that perspective in mind, that is the end goal, that we are hoping to develop therapies for some of these really devastating diseases. So it's always useful to have that perspective. And I always welcome it in a meeting, and I'm very glad to see it being represented.
Yeah, I think a lot of the insights have come that have led to these attempted treatments, and we still have to tweak how to get that impact there. But yeah, there's definitely a lot of movement in that direction, which is great. Gabriel, would you like to go?
Yeah, absolutely. I think that the chairs have done an amazing job at selecting the speakers and the topics. It's been really, really exciting. And I'm particularly very interested in an integrative perspective. So when people engage in studying, how exercise or sleep or peripheral signals affect the immune system and the brain, there is historical tendency of scientists to try to divide an organism or biology into approachable sections, but the truth is that every system which is arbitrarily defined talks to each other, and I find it fascinating, how exercise has an influence on the immune system, which in turn has an influence on the working of neurons and glia, and how epithelial also has an influence on the working of those two systems. So I think this is really fascinating, and the talks that have been given on those areas have been really engaging and insightful.
Great, yeah. And Sophie?
Yeah, I agree with all of you. I personally had two standouts, not to be specific, but like the keynote speaker, Akaiko Wazaki, I thought her data on like the opening of the blood-brain barrier after infection and like the potential to see T-cell priming was like super cool, and like understanding how you could potentially modulate that. I thought that was super exciting. And also I kind of come from a research background where I looked at like prion-like spreading, so that thought is always in the back of my mind, and I was thinking, okay, so it kind of reminiscent of those like intranasal inoculation studies of like PFFs, the synuclein, and wondered whether like these other kind of protein aggregates can also prime T cells and open up the blood-brain barrier and that kind of thing. And yeah, I think that's why this conference is so unique and such a great opportunity, because it's bringing those two fields together to kind of like initiate those talks. But I also thought, so I really enjoyed the talk by Jennifer Yokoyama, because I just kind of thought there's like this increasing knowledge that peripheral responses are happening in neurodegenerative diseases. And I guess I've always kind of thought of them like as a very central nervous system, and they are like diseases, but you can have like peripheral responses going on that reflect on what the disease state is. And I thought that was super like exciting, quite revolutionary for potentially for like biomarkers or early diagnostics. So yeah, they were like my takeaways.
Yeah, definitely. And yeah, I know that's one of the biggest challenges. How do you know what's going on when you can't really get into the brain? So that could be a really, really great way to get an insight. Great. So let's dig into a little more detail into what kinds of stories you've got your eye out for at the meeting to feature in upcoming issues of your journal. And those can be, let our audience know if those are more molecular mechanism or clinical advances or both. So yeah, just give them an idea of what you've got your eye out for. Maybe Gabriel, you want to start off this one?
Yeah. As I said before, we are a very general biology journal. So pretty much like anything in this set of conferences is within the scope of PlusBio. But because of our audience is a general audience, I think that any integrative story, it's a very strong candidate for our journal. Any story that combines different approaches or tackles a single biological problem, approaching it from two different systems like the immune and the nervous system are very good fits for PLOS biology.
Great. Swapna, would you like to go next?
Sure. So I guess, again, no surprise that we are looking for studies that have strong clinical and translational relevance. And particularly for this field, there's certainly for neurodegenerative diseases, there are already therapies that are in the clinic or on their way to the clinic. So of course, trials involving those therapies would be an obvious fit. But for many of the other talks that we've heard, I think fundamental insights into disease biology that yield actionable mechanisms for therapeutic intervention, I think those would also be studies that are within the remit of the journal. I think it's a little premature to expect direct therapeutic insight, especially for some of the more basic work that we've heard. But certainly, I think any studies that shine a light upon fundamental disease processes and offer at least strategies for how these might be therapeutically modulated, I think anything like that would be a good fit with the scope of med.
Great. Sophie?
Yeah, so Nature Communications is quite broad. So I'd say everything here is in scope essentially. So we publish like molecular, kind of more mechanistic underpinnings of like neurodegeneration, to also kind of like Alzheimer's disease modeling papers. So I would say if in doubt, just submit. We encourage all types of submissions all over the field of neuroscience. So yeah, we're quite broad.
Great. And Tim.
Yes, so as I mentioned before, Journal of Cell Biology, most of our work tends to be more mechanistic, more cellular, molecular, and so those tend to be the types of things that we're looking for. But honestly, we have a pretty broad scope in terms of what constitutes cellular biology. If you're looking at how groups of cells or cell-cell interactions influence pathogenesis or pathology progression, things like that, even if there's not sort of necessarily looking at the molecular underpinnings of that, that's still appropriate for Journal of Cell Biology. But it is a little bit more constricted in the sense that the more translational stuff would be interesting to us if it has a cell biological aspect to it, but purely transitional stuff won't necessarily be a good fit for JCB. But again, we cast a pretty wide net in terms of what we could constitute cellular biology.
Great. So for those who are looking to submit their work for publication, whether that be currently or within the coming year, what would your best piece of advice be for them in submitting? And that can be either something that they should definitely do when they contact you or when they make their submission, or maybe something that they definitely should not do. So whichever you'd like to focus on. Swapna, you want to start this off with this one?
Sure. My typical advice would have been look at what the journals published, and that should give you a sense of whether your work is a good fit or not. Unfortunately, for a newer journal like us, it's not always possible to find whether your work is a good fit for the scope. So I would strongly encourage people who are interested to actually reach out to us via a pre-submission inquiry, or just, it doesn't have to be even that formal. You could just shoot us an email to see whether your work is in scope for the journal. I think that would be the easiest way, and that really would be my advice at this point. If you are interested, we're easy to find. We very much want to talk to our authors, and quite frankly, we miss that. It's difficult to recapitulate virtually. So we would love to talk to you if you have work that you think might be of interest to Med.
Great. Yeah. So you don't necessarily have to be ready to submit the full application.
I do want to say that it doesn't have to be a complete story. If you have something that you think could be potentially ready for the journal in the future, that's fine too. We're still happy to talk about it and maybe give you a better sense of what we're looking for, so that can help you when you actually get ready closer towards submission.
Great. Tim, you want to go?
Sure. So I don't believe that there's any secret formula to getting your paper into any journal. Honestly, I think all journals are looking for the best possible work that they can publish. So if you've got a really solid, interesting study, then you're starting from a firm, grounding. I would say that, one of the things that you do have to remember when you're writing your paper is it's really important to remember that once you start, you have to make this transition from doing your research and thinking about your project from the perspective of what experiments I have to do, to translating that to becoming a storyteller and sort of putting your paper together in a way that makes logical sense to any given reader, which includes the editors, right? When you first submit it, your very first reader is going to be the editor. So you've got to put your story in a way that's convincing that person why they should be interested in that, why that story is really cool, how it moves the field forward. I think it's really important that you don't try to go overboard. I think there's occasionally you'll see people who do what I call data dumps, and they'll just throw every piece of data into the paper that they can. And I recommend not doing that, just because it can dilute the message. You want to have a clear, concise message that you can convey to someone in 1 or 2 sentences, and have all the data that is necessary to support that main conclusion, but not anything extraneous. And also make sure you have all the data to support that conclusion. And I think that goes for any journal, not just Journal of Cell Biology. I think you are catering, when you're trying to get your paper into a journal, the first reader, as I said, is the editor. And so explain to me why this story is really cool and what's so interesting about it and how it moves the field forward. And that's the first step, I think.
Yeah, I think that's a really good point, because when you're doing the research, you're sucked so into the details that sometimes you forget to take that step back and communicate to other people why it's so important or cool, because it's so obvious to you, but no one else has the perspective that you do. So
I do think that some labs do it better than others in the sense that, when I was a grad student, I was taught to do experiments and how to think about what my next experiment would be and, my end goal, but no one ever really taught me how to go about writing a paper. Where do I start? How do I put my ideas together in a cohesive manner? And I think that's something that's really important to learn.
Mm-hmm. Great. Yeah. And I've also heard other editors in the past on that same note say, if you give it to somebody who's not directly in your lab to read, does it make sense to them, to somebody who's not directly in your lab and doesn't already know the background? Great. Sophie?
Yeah, they were excellent suggestions from both Tim and Swapna. To kind of go on from Tim's point, I'd say a cover letter is quite a useful tool. So if you're not quite getting across your kind of what your advances in the paper, you can use the cover letter to really kind of delineate what the specific advances and emphasize anything to the editor personally that you probably couldn't put into the paper. So I think they're quite a nice tool just to kind of get your message across and say anything that you want to say privately.
Great. Yeah. And Gabriel? Yeah,
well, I think that they already said the three advices that I would have told them. Approach your editors. I think that we work for you and we want to make your experience as productive and as pleasant as possible with any of the journals for which we work. I also believe what Tim said about being a storyteller. Sometimes the work in the lab is a little bit messy and you move back and forth and you change hypotheses and your results can't be clear and you have to clarify. But that's not, you don't have to write your paper as chronologically as experiments were done. You have to work on putting your results together in a way that is logical. And finally, I personally find the cover letters very useful. And my invitation is that you as an author are very candid about what pieces of your study are confirmatory and what pieces of your story are novel. And that information should be conveyed in the paper itself, but a good, strong, solid cover letter that is not just a repetition of the abstract, but is your opportunity to engage in a conversation with the editor, tells those aspects as well.
Great. Yeah. And I would like to dive into that cover letter versus abstract difference in just a moment here. I'd like to remind our audience, you can submit your questions into the Zoom chat. We're going to open up for audience questions shortly. So get those questions in. So one final question for the panel. What is the difference then between what should go in your abstract and the body of the paper versus a cover letter, and what makes a good cover letter? Maybe Gabriel, if you want to expand on.
I think the abstract is the summary of the paper, and it has to be structured in a way that it presents a context or a background. Then the abstract itself presents the content of the paper, briefly summarize the most important results, and at the end of the abstract, 1 or 2 sentences for the conclusion. So there's context, there is content, and then there is conclusion in the abstract. And it is mostly targeting your broader audience. It is targeting your colleagues. Many of your colleagues will only read the abstract, and many of them will decide whether they dig into the paper based on the abstract. So the cover letter, in my opinion, is a conversation that you have with your editor. And I don't think that a poor cover letter will have your paper rejected, but a good cover letter might make your paper stand in the eyes of the editor. And sometimes authors include in the cover letter very useful information that it's not pertinent to put in the abstract. For example, I mean, I can tell you a couple of examples of cover letters that historically had been like helpful to me as an editor to decide what to do with a paper. Sometimes, you know, a different group has reported something that is very similar or that have reached your conclusions, but in that previous paper, the authors pretty much made a guess, and they didn't have strong experimental evidence to support those conclusions. But if I, as an editor, just read the abstract of the other paper and take it at face value, I may think that your results are not sufficiently novel. But in the cover letter, there's an opportunity to say, we actually demonstrate what the other paper claimed, but was only a guess, right? So we do provide the data to support these conclusions. And that's a conversation that you can have in a cover letter with your editor. Another example is, you know, like if your paper has been rejected previously and you understand the reasons for the rejection, you can tell to the editor of the next journal, you know, my paper has faced these hurdles before, which are fair or unfair according to your point of view, and expose why, you know, the previous journal, despite the caveats of your paper, or this journal, you know, should consider your paper. So explaining more the limitations of the study in a candid way to discuss with your editor.
Okay, great. Anyone else want to add to that?
I'd certainly agree entirely with what Gabriel said. One thing that I think I would add, or two things I would add. First, when it comes to the abstract, is just making sure that your abstract conveys what, you know, the main advances of your paper are, but not overreaching. Particularly when it comes to disease studies, I think sometimes authors attempted to make logical leaps into how this applies to disease when they haven't actually tested that. So I think it's important in your abstract not to go too far. That's not to say that you can't draw, you know, parallels between what you're looking at and known disease pathways. Just be sure that what you're saying in your abstract is actually supported by the data in the paper. And when it comes to the cover letter, one of the things that I find useful is it's, besides being a place where you can sort of speculate a little bit more and provide a little bit more context that you can't necessarily do in the paper, as Gabriel said, I think also, you know, when you can also use that as a place to indicate, you know, potential reviewers that you think are good referees for your paper, or people you'd like to exclude. And I think that's useful information, even if the editors don't necessarily use that information. It helps tell me where you think your paper fits within the literature. The people that you choose as potential referees tells me, okay, you think this fits here, and this is how it applies to other studies in literature around this group of work. So I also find that kind of useful from cover letters. But beyond that, I completely agree with what Gabriel says.
Okay, great. And Swapna or Sophie, do you have anything to add?
Yeah, I just had one point. I do agree that a good cover letter helps, but a bad cover letter doesn't necessarily hurt. So it's not absolutely essential. And I know, you know, we have heard from authors who are worried about this and that there really isn't much to be concerned about. Personally, what I've also found useful is, especially in fields which are, you know, there's a lot of debate, or there are certain points which haven't been resolved, or maybe there are some contentious questions and there are no easy answers. I think providing some of that context in a cover letter can be useful, because sometimes, you know, as an editor, I may not necessarily know the ins and outs of a particular hypothesis, or whether it's accepted or not accepted, and there may be a lot of conversation about it that I may not have all the details of. So I think a cover letter is a good place to just sort of briefly sketch out that maybe there's debate in this field about a particular question, and our work seeks to address that in the following manner. So that kind of context of where the field is going and where your work fits into the broader questions that the field is considering, I think that can also be quite helpful.